Helicobacter pylori normally exists in a spiral form. Under environmental stress, its shape gradually changes. The cell shortens, loses motility, and becomes rounded. It eventually transitions into a coccoid form, a state associated with increased environmental resistance. Mechanism of Helicobacter pylori coccoid transformation, antigenic changes, tendency to disappear, and reversion conditions. Helicobacter pylori typically exists in a bacillary or spiral form, but under stress conditions, it adapts by entering a coccoid form, which is a VBNC, viable but non-culturable state. This adaptation mechanism is believed to be involved in relapse after eradication therapy and diagnostic evasion. The following is an explanation based on scientific findings, primarily from recent reviews and papers. 1. Mechanism of coccoid transformation. Coccoid transformation is not cell death, but an active survival strategy. The main inducing factors and molecular processes are as follows: inducing stress conditions, nutrient deprivation, increased oxygen concentration, pH changes, alkaline or acid stress, high temperature, and prolonged cultivation. Exposure to sub-MIC, sub-minimum inhibitory concentration, of antibiotics, examples, amoxicillin, clarithromycin. Oxidative stress, example H2O2, and inflammatory environments. Process of morphological change. The form transitions from spiral to U-shaped or C-shaped intermediates to fully coccoid form. Electron microscopy reveals an expansion of the periplasmic space, separation of the outer and inner membranes, and remodeling of the cell wall, peptidoglycan. Flagella may also wrap around the cell. Molecular mechanism, a key focus in recent years. The type 1 toxin-antitoxin system, particularly the AAP1/ISOA1 pair, plays a central role. Normally, ISOA1 in antitoxin, antisense RNA, inhibits the AAP1 toxin mRNA, suppressing toxin production. When ISOA1 is reduced or suppressed by stress, such as oxidative stress, the AAP1 toxin is expressed. This toxin acts on the inner membrane to rapidly induce coccoid transformation, but the cell membrane remains intact, and there is no significant drop in membrane potential or ATP levels, meaning it is not accompanied by cell death, based on cryo-EM observations. This improves antibiotic resistance and environmental adaptation. The involvement of cell wall synthesis-related enzymes, penicillin binding proteins, PBP1, 2, 3, and cell shape determining proteins, CSD1 to 6, MREB, and CCMA has also been reported. This process is a programmed conversion for survival, not merely aging or degeneration. Although some forms are degenerative, many coccoid forms retain viability. 2. Disappearance or reduction or alteration of antigenicity. Accompanying the coccoid transformation, the surface antigen profile changes significantly, resulting in the loss or reduction of certain antigenicities. This leads to immune evasion and diagnostic difficulties. Specific changes. A 1996 study by Benessa, Infect Immune, confirmed that antigens change along with ultrastructural alterations during coccoid transformation. Specific surface proteins, example 160 kDa, 120 kDa, VAC-A related, and 35 kDa surface proteins disappear or decrease. Conversely, some high molecular weight antigens are reported to be expressed specifically in the coccoid form, changing the overall antigen pattern. The sensitivity of tests such as the stool antigen test decreases due to the downregulation of surface antigens. Some virulence factors, example CAGA, VACA, are also downregulated, leading to reduced cytotoxicity. However, they are not completely lost and maintain a low level of expression. Impact on immune response. The immunogenicity of the coccoid form is reduced, making it easier to evade the immune system, though it does not disappear completely. There are cases where IgG antibodies recognize both forms, especially in children, where coccoid-specific antibodies may be high. This is thought to promote chronic persistent infection and relapse. 3. Conditions for reversion, resuscitation. The coccoid form is reversible. When the stress is removed and conditions become favorable, it reverts to the spiral form, recovering its culturability, infectivity, and antigenicity. In vitro reversion conditions. Transferring to fresh media, nutrient supply, increases ATP levels and recovers urease activity. Reversion to the spiral form has been observed approximately 18 hours after subculture. Resuscitation promoting factors such as RPF proteins derived from other bacteria and heat shock proteins, example HSPA/B, may be involved. In vivo reversion. This is most frequently confirmed in mouse models, BALB/C mice. When 20-day-old coccoid forms are administered, they colonize the gastric mucosa, revert to the spiral form, and induce inflammation, histopathological changes. They naturally revert in the favorable environment of the stomach, appropriate pH, nutrients, mucus layer, causing sustained and relapsing infections. Reversion requires membrane integrity and residual metabolic activity, such as DNA synthesis and mRNA retention. Not all coccoid forms are capable of reversion. Degenerative forms are irreversible. Clinical significance. Cause of eradication failure and relapse. Coccoid forms have high antibiotic resistance and are difficult to detect via standard culture, making them prone to remain after treatment. Diagnosis. PCR and electron microscopy are effective. Treatment strategies that account for coccoid forms, such as re-evaluating after a period of antibiotic suspension, are heavily emphasized. This is a summary based on current scientific knowledge, primarily from reviews and papers published between 2020 and 2025. Research is ongoing, and the specific details of the TA system are relatively recent discoveries. Please consult a medical specialist regarding individual clinical cases. Main references, excerpts. Sadeklu, 2025, Coccoid H. pylori, a key driver, etc. Ierardi, 2020, The Puzzle as Coccoid Forms, etc. Benessa, 1996, Changes in ultrastructure and antigens, etc.